Good morning and a very happy new year to you all. Today we'll talk about biological sequences and their application through bioinformatics. Bioinformatics increases the understanding of biological processes and it focuses on developing and applying computationally intensive techniques. The foremost and extensively used technique or method in bioinformatics is sequence analysis, which subjects to use of DNA, RNA, or a protein sequence and to understand their feature, function, structure, and evolution. Sequence analysis find wide range of application in understanding the evolutionary processes on the onset of diseases and how they are involved in disorders. They help in the identification of conserved sequences like restriction sites, motifs patterns, and many other intrinsic features. Sequence analysis also help in gene prediction and comparative genomics. It's an important step of um, phylogenetic profiling and structure prediction. Also, they could help us in identification of sequence differences and variations, that is, mutations, and understanding single nucleotide polymorphisms. Alright, so how do they help in rearrangement? And what are rearrangements? Rearrangement is a structural alteration in the chromosome which usually involves a breakage and reattachment of a segment of chromosome resulting in an abnormal configuration, such as inversion deletion, and so on. The main role of gene rearrangement is to create diversity, and they could play a major role in evolutionary processes, mutations, and disorders. Here are we have listed a couple of diseases where the gene rearrangement occurs. For example, we have leukemia, where at the early beta cell development, the IGH gene rearrangement occurs. In aggressive lymphoma, the MYC gene rearranges. So, how do you arrange the rearrangement in a sequence? You could use a simple BLAST program to identify at what location the rearrangement or the changes have occurred by comparing a normal sequence against a diseased one. Also, there are specific programs which have been designed to study the DNA rearrangement or the genomic rearrangements across the genome. One such program is SPRING, and there's another one called as GRIM, GRIMM. You could please make a note of it. Also, gene rearrangements could be used in understanding gene prediction. They could be identified using multiple sequence alignment, and they could also be of phylogenetic inferences. So, moving on, let's know about conserved sequences. Conserved sequences are similar or identical sequences across the genome within an organism or across the species. And like we have discussed before, these are this is intrinsic feature of an biomolecule, and this includes restriction sites, domains, motifs, patterns, and so on. So the conserved sequences could serve as evidence for structural and functional conservation and help us in understanding the evolutionary relationship between the sequences. Because of this, they give a greater scope for researchers and reduce the time consuming experiments. They could be sequences which are conserved, yet they might be lying in the non-coding DNA and are not much understood and could have high functional value. They're also interesting because they might have circuits of regulation, not exactly the coding gene, but the regulatory sequences might lie in the conserved region or the non coding DNA. They could unveil the effect of mutation, help us in building the phylogenetic tree, and understand cross species conservation. They might also help in designing new biological systems and predict functions of new proteins and hypothetical proteins. Now, hypothetical proteins are those whose existence have been predicted as a result of genome sequencing. However, there is no evidence that they are expressed in vivo. So, moving ahead, promoters are those regions of DNA that initiate transcription of a particular gene located towards the 3' end of the antisense strand upstream, called as the template strand or the non-coding strand. They're about 100 to 1000 base pair long, and in prokaryotes, we have the promoter region at the -10 sequence, which is called as the Pribnow box, and at the -35 sequence, where the RNA polymerase recognizes and binds. So, in eukaryotes, we have this region at the 30 base pair data box and the 100 base pair where and the promoter region where the RNA polymerase binds. Now, lying adjacent to the gene in question and position designed relative to where the transcription begins, the promoters have the potential to regulate gene expression. So, promoter prediction is an important step or traditionally the first step to gene prediction. And by inducing different intracellular and extracellular environment, we could study the effect of the environments on gene production and how the gene reacts. Also, by inducing different drugs at the promoter region, we could check the gene expression pattern, and this field is called as the pharmacogenomics. Role of promoters also could be studied in evolutionary biology. And for example, if one wants to study how humans have evolved from chimps, it would be interesting to study if there has been any evolution or mutation in the promoter or the regulatory region, or is it because of an evolution in only the coding sequences that humans have evolved from the monkeys. Promoter malfunction also have led to, and there are well-known diseases linked to it, like asthma, beta thalassemia, which is an inherited blood disorder. We have Ribstein-Tabby syndrome. The patients have short stature and severe learning difficulties, and which is an increased risk of cancerous and non-cancerous tumors and leukemia and lymphoma. So these are some of the applications of promoters and they scope for research. These are a couple of tools using which we could predict the promoters in a sequence. Intergenic regions. Intergenic regions, as we all know, are the regions lying in the non-coding part of the DNA or the regions in between the genes. They do not participate in protein synthesis, nor do they contribute to the phenotype of an organism. But are they just the non-coding junk DNA, or do they play a role in the regulation of a gene and its expression, apart from just giving stability to the genome. The non coding DNA might have certain regulatory sequences or patterns which might be of importance and are not much studied. For example, let's talk about genetic silencing and imprinting. Genetic silencing is the ability of a cell to prevent the expression of a certain gene. Unlike knocking down, the gene knockdown, which we all know, where the expression of a gene is reduced, silencing is a process where um, the expression of a gene is completely turned off. And this turning off is called as imprinting. So, imprinting is an epigenetic phenomena which could occur because of DNA methylation or a histone modification, most probably in the non coding part of the DNA before the gene. So, look at this example. Generally, a child inherits one copy of gene from the mother and another copy from the father. In case one of the copies that he has inherited from the father is a mutated or a diseased allele, and the normal allele that he has inherited from the mother is imprinted or turned off, there is every chance that the child would, for 100%, have a high risk of developing the disorder. So, this is called as gene silencing and imprinting. And the gene enhancers or silencers are those part of the junk DNA or the introgenic DNA which control the genetic silencing and imprinting in an organism. Moving on, there are other regulatory regions like response elements and enhancers, silencers, or even the promoter region and other patterns and motifs, cis regulatory elements which might lie in the intergenic regions. Now, here we list few transcription factor binding sites and tools to identify them in the DNA. These are databases for promoters. EPD is a eukaryotic promoter database at EMBL. PlantKit and PlantPromDB is a database of plant promoter sequences. Saccharomyces cerevisiae has a specific promoter database called SCPD. These are a list of tools for your convenience to find patterns and motifs. Based upon your requirement, you could browse through them and choose the required tool for your analysis. So these are specific to cis regulatory elements, and these are again some databases for sequence motifs. Alright, so next we have the restriction sites. Restriction sites are palindromic sequences which are 4 to 8 base pair long generally, with a recognition site where a restriction enzyme binds and cuts. Here is an example of a couple of restriction enzymes from the respective organism, the recognition sites. Right. So restriction enzyme find a great application in recombinant DNA technology, DNA therapy, biopolymers, cloning, DNA fingerprinting, and also they are linked with certain genetic diseases and could be used in identification of them, like sickle cell anemia, Hodgkin's disease, Duchenne muscular dystrophy. Right. NB Cutter is a tool. It's an online tool where, let's say you are performing a recombinant DNA technology. Instead of spending hours in the lab trying to identify the splice sites or the restriction sites of your sequence, and experimenting with different restriction enzymes, you could give your sequence in this tool, and it would give you the restriction sites and the possible restriction enzymes that could work on your sequence. So this would save a lot of time, and you could directly choose the enzyme of your interest and proceed with your research. Now, Vector NTI is it's a package. It's a completely integrated suite of various tools, including sequence analysis, and it also has a very interesting tool for a software for designing recombinant DNA. I'll give you an example of biopolymers that I had worked on. Using Vector NTI suite, what we did is we chose the PHB genes from alkali genus. Now, PHB genes are the genes that produce biopolymers in a very low amount in alkali genus. So we took these genes from alkali genus and we inserted them into the starch gene of Ipomia batata, that is the sweet potato, and created a recombinant DNA. So this could be exploited at a large scale industrially to produce biopolymers and to replace the synthetic plastics that cause the environmental pollution. So that is one example how recombinant DNA finds application using bioinformatics, and Vector NTI is a very good package for that. Right, so let's move on to our next topic, single nucleotide polymorphism. Single nucleotide polymorphisms is a mutation that occurs in a single base at one point or one nucleotide, and on an average, one SNP occurs in every 1,000 base pairs. This is a permanent feature of a genomic DNA and could be helped in studying genetic associations and diversity across population, which the subject is called as population genetics. All right, they might also be linked to alleles of health-related genes or recessive genetic diseases. So they find application in medical field as well. Now, a few of them could also have evolutionary advantage, and less than 1% of the SNPs actually impact the regulatory element. All right, like I said, SNPs find a great scope and application in pharmaceutical areas and companies. They find an application in pharmacogenomics and personalized drugs. Now, personalized drugs are drugs that are given on gene test. Now, this is the future of our pharmacogenomics, or this is our future of bioinformatics. Based on the gene test, drug response data would be collected and genetic profiles would be created. These genetic profiles would be used to prescribe drugs to you to avoid adverse reactions. So this is the application of SNPs, which holds a great future, and this is how it could be applied in pharmacogenomics. Right, so OMIM is a database of NCBI which holds about more than 2,000 genetic diseases and information on them. This is specifically designed for physicians and for their research and for clinical studies. DBSNP is a tool which is again from GenBank NCBI, which will help you to identify SNPs in your sequence. So you could use this tool, and the most commonly used tool, to analyze the SNPs in your sequence, and it would also help you to locate directly where these points are located on your genome. Right? So HapMap project is again based on SNPs. This tests linkage between the SNPs across various subpopulation. The goal of this project is to define the patterns of genetic variation across human genome and to guide selection of these SNPs efficiently to tag common variants. All right, next comes the gene prediction. Gene prediction is a process of identifying the regions of genomic DNA that encodes genes and their related regulatory regions. And this is the first and the most important step of understanding a sequence genome. And we should not forget that the functionality of a gene and its prediction is based only on the sequence. So there are different techniques or approaches of gene prediction. We have extrinsic approaches, we have intrinsic approach, it's also called as the ab initio approach, and also comparative geno approach. So let me describe a couple of lines about each one of them. Extrinsic approach, or the evidence-based gene finding systems, are entirely based upon the known sequence of mRNA or protein sequences, or the reverse translation or transcription. It searches a target genome for matches, and the major drawback about this approach is it needs extensive sequencing, and we know that not all the genes are readily available. So that is the major drawback. BLAST generally is designed for this purpose. Apart from that, there are specific databases which contains transcripts and translated sequences from different species. That is the RefSeq and Ensemble system is two such database, I would say, which contains sequences for extrinsic approaches. Right. So the next one is the ab initio approach. We can also call it intrinsic approach. This is a systematic. This is a more systematic approach, and it uses the patterns and conserved sequences of a biomolecule. The genomic DNA sequence is systematically searched for signs of protein coding genes, that is with the help of, like I said, conserved regions and patterns and motifs like open reading frames, the promoter sequence, the poly A tails, splice sites, CPG islands, and so on. There are specific tools which are designed based on ab initio approach: GeneMark, Glimmer, GenScan, Snap, Gene Finder, and there are many more. Right? Combined approaches are approaches which includes both intrinsic and extrinsic method, and it's always better, and it is always good to use combination of approaches to reach consensus. So combined approach uses both intrinsic and extrinsic approach, and it uses EST alignments and protein profiles to increase the accuracy of a gene prediction. Now next is the comparative genomic approach for gene prediction. This is based on the principle that the genes and the functional elements in a sequence in a biomolecule, they undergo mutation at a slower rate compared to the rest of the genome. Thereby, these sequences are more conserved. So, based upon genomic comparison between related species, it is easier to deduct the evolutionary pressure for conservation using comparative genomic approaches. The tools that use comparative genomic approach for gene prediction are SLAM, SGP, NSCAN, TwinScan. There are a few more: Projector, GeneWise, and GeneMapper. And comparative gene finding actually gives high quality of annotation from one genome to another. So, though there are different techniques of gene finding or the gene prediction programs techniques are available, as I mentioned earlier, it's always good to use combination of tools to compare that the results that you're receiving or are reaching at the end has a consensus, and that would give you a better result and a strong proof for your analysis. All right, these are a few more list of gene prediction software. All right, so that is all about it. I hope you had understood a bit. If you have any questions, any doubts, you could please write into us. Thank you so much for patiently listening to the presentation. Thank you.